So today we're going to be talking about the electron transport chain, okay? So we know about respiration, right? Basically the process through which we are utilizing or producing the energy from the food by their oxidation, right? So it basically consists of the glycolysis, it consists of the Krebs cycle, and the very last, it consists of the ETC, okay? Krebs cycle and the ETC, or the electron transport chain, okay? So in this glycolysis and the Krebs cycle were the production of the NADH and FADH2, right? And those NADH and FADH2, why we say that this NADH is producing 3 ATPs while the FADH2 is producing 2 ATPs, right? So how are they able to produce the ATP? How are they able to do this energy production? Why are we saying they're producing 3 ATPs and why are we saying 2 ATPs? That is basically these two are being utilized in the electron transport chain, okay? Where basically what happens in the electron transport chain is that they both get oxidized, okay? And then they undergo series of oxidation reduction reaction, and as a result of that, what happens is that we get the energy for the the production of the ATP, and basically at the also what happens is that there is the final acceptor of it, which is referred to as the oxygen. I'm going to discuss all of that, energy production one, and how the final acceptor oxygen helps works out in the ETC. We're going to discuss all of that in detail. So basically, you need to know this basic concept, okay? First of all, there's glycolysis and the Krebs cycle, which are producing NADH and FADH2. These two are going to be utilized by the ETC, and they're going to be oxidized, and they're moved through series of oxidation reduction reaction, and they're going to be their final acceptor is going to be the molecular oxygen, and their electrons are going to be utilized for the production of ATP. How are we going to discuss all of that? Okay, moving on with our answer. When you discuss the components of ETC, you should first, before I jump into how the passage of electron flow happens in there, how that basically happens, we're going to discuss what are the components of the ETC. Remember this one thing, as the name suggests, it is electron transport chain, means electrons are being transferred, and as electrons are being transferred, that electron transport results in the production of ATP, and they are also utilized in the production of water. We're going to discuss all of that in a little bit, but that is the basic concept about ETC. So let's talk about the components of the ETC without further ado. Okay, so the first component that you're going to be discussing about is the complex one. When you hear the word complex one, what do we refer to as? The NADH dehydrogenase complex. What is it? The NADH dehydrogenase complex. What do you mean by dehydrogenase? We're going to discuss that in a little bit, but remember this dehydrogenase word, okay? Next, the second component of it is going to be the FADH dehydrogenase complex, which is going to be the complex 2, okay? This is going to be FADH dehydrogenase complex. Same dehydrogenase is going to be here too. Third one is the coenzyme Q. Okay, what is it? Coenzyme Q. Fourth one is the complex number third, which is the complex third, which is cytochrome reductase complex. Okay, cytochrome reductase complex. What is it? Reductase. Now from the name you can already guess what is it going to be doing. Moving on further, we have cytochrome C. Okay, it's going to be cytochrome C. This is not a complex like the cytochrome Q, coenzyme Q. They both are not complexes, okay, like the other ones we have. We're going to discuss their role in a little bit. C1 is this the fourth complex, or the last complex of it, and that is basically what we call as the cytochrome oxidase. What is it? Cytochrome oxidase. One is reductase, the other is oxidase, okay? One is NADH dehydrogenase, the other is FADH dehydrogenase. Your name is like now clear, okay? Complex 1, NADH dehydrogenase. Complex 2, FADH dehydrogenase. Complex 3, cytochrome reductase. Complex 4, cytochrome oxidase. In between them is the coenzyme Q and the cytochrome C, which roles I'm going to be discussing in a little bit. But remember this, they are basically mobile electron carriers. What are they? They are mobile electron carriers. What are they? Mobile electron carriers, because they're going to be carrying and transporting action of the carrying the electrons. Okay, so you have NADH dehydrogenase, FADH dehydrogenase, coenzyme Q, cytochrome reductase, cytochrome oxidase. First write all the complexes down, then write the coenzyme and the cytochromes at the very end when you're discussing the different components. Okay, moving on with our answer, moving on further with our topic. Now we're going to be discussing the diagram of this ETC. We have complex 1, 2, 3, and 4, and in between we have the coenzyme Q and the cytochrome C here, right? We have discussed all of that in detail. We are going to be discussing them in detail right now. So let's move on with our answer. First of all, what is this? Complex number 1. What was the complex number 1? NADH dehydrogenase. Remember this. Now I'm going to be telling you this. Whenever you hear the word dehydrogenase, means it is the removal of the hydrogen. So what do you mean when you hear the word NADH dehydrogenase? Is that there is going to be removal of hydrogen from the NADH. Okay, so what happens is that NADH plus H positive, it's written like that, it will be transformed into NAD positive plus 2H positive plus 2 electrons, because to balance out the positivity we have the electrons there. They're also going to be released. That is what dehydrogenase. Same if it is FADH2 dehydrogenase, it is going to be the same way. It is going to be that FADH2 is going to get transformed into FAD positive and 2H positive and the 2 electrons. Okay, this is what we mean by dehydrogenase. So both the complex 1 and the complex 2, they're both responsible for the dehydrogenation of the NADH and the FADH. So from NADH, what will happen in the NADH dehydrogenase complex? There is going to be the release of the electrons. Along with that, hydrogens are going to be released. Those electrons will move to the coenzyme Q. What is it going to move to? Coenzyme Q. If FADH2 is to be oxidized through ETC, it also hands over its electron to coenzyme Q. Okay, if there is FADH2 available, if it has to be oxidized, then it will also move its electron to coenzyme Q. Okay, so both NADH2 dehydrogenase complex and FADH2 dehydrogenase complex releasing the electrons will be passed to the coenzyme Q. Moving on further, towards the third complex, we have the cytochrome reductase complex. Okay, what is it? The cytochrome reductase complex. Now, this cytochrome reductase complex, what is it going to do? It will be handing over the electrons from the coenzyme Q to the cytochrome C. Okay, what is it going to do? It is going to be handing over the electrons from the coenzyme Q to the cytochrome C, which is another mobile electron carrier. Okay, it is another one mobile electron carrier, and it is going to carry the electrons towards the complex number 4, which was what? This was cytochrome oxidase. Okay, what was it? Cytochrome oxidase complex. And what this oxidase complex is going to do, it is going to basically cause the electrons to move in like that, and it is going to basically, those 2 electrons, they're going to be adding up with the 1/2 of oxygen molecule, and 2 hydrogen ions are going to enter to forming the water molecule. Okay, as a result, I told you at the very beginning at the ETC, the final molecule, the acceptor of the final acceptor is going to be what? Or the oxygen. Okay, that is what I'm showing you at the very end. It is going to accept the it is going to accept the electrons that were being produced as a result of the dehydrogenation of the NADH and NADH and FADH2. Those two electrons that are being released by them, they're being accepted by the oxygen to produce the water molecule. Okay, that is one thing that happens during the pathway. But remember one thing, that this is the energy around this pathway is in this direction. Okay, now what do you mean by when I say this? Means this one, this complex 4 is the lowest energy one, while the complex 1 is the highest energy one. When the electrons are being moved from one complex to another and it is going down the gradient, down the energy, these electrons are basically going to be releasing the energy, and that energy is going to be utilized for the production of ATP, which I'm going to be discussing in a little bit. So as you move around down this gradient, it is being utilized for the production of ATP, and I'm going to be discussing that how in a little bit. So till here there should be no issue in the ETC. Okay, so there is no direct production of ETC going to happen in the ETC through the ATC. There's going to be a pathway which we're going to discuss in a little bit, but this is what happens in the ETC. So you have ETC, which is basically going to be oxidizing the NADH FADH2, and as it is move them through different oxidation reduction pathway, and as a result molecular oxygen is going to be the final acceptor at the very end of this pathway. We have different components of the ETC, complex 1, 2, 3 and 4, and I told you a way of remembering them. The first one is the NADH dehydrogenase, the second one is the FADH dehydrogenase, cytochrome reductase and cytochrome oxidase. Okay, so that you should remember, just NADH and FADH2 here, reductase and oxidase here. All right, and we have coenzyme Q, coenzyme C, and they are just mobile carriers. Okay, until here we are done. Moving on towards the next topic is how this basically ETC is able to produce the ATP. We're going to discuss that. Okay, so basically what happens is that along this, what happens is a process of chemiosmosis. Okay, what is it? Chemiosmosis. And due to the chemiosmosis process, the ATPs are being produced. Remember one thing about ETC is that you have mitochondria, right? And there's the mitochondrial matrix is the presence of the Krebs cycle, but there's the inner membrane of the mitochondria, which is consisting of the Christie, is the region where the ETC happen. Okay, where the chemiosmosis, which I'm going to discuss here, is going to be happening right there. So this is the location of the ETC where it basically takes place. Moving on towards the next topic, okay, that is the chemiosmosis, which is related to the ETC. Once you get to know that, we're going to be discussing. This is very easy. Okay, so what happens in the ETC? In the ETC, you have the complex 1, you have the complex 2, you have complex 3, which I'm not going to discuss later on what are they doing. We discussed that right now in a little bit before. We have the coenzyme Q and we have the cytochrome C. The electrons are going to be flowing from one to this. We have flowing happening and they're going to be entering here. And finally, they're going to be accepted and they're going to be utilized for the production of water. Okay, but what happens is that they're going down the energy gradient. They're going right. They're going down the energy gradient, and the energy, as they're going down, the energy is being released by the electrons, which basically this energy being released by the electrons is utilized by the hydrogen ions. Okay, what happens? The hydrogen ions, what happens to them is that they are being pumped out, okay, from the inner mitochondrial matrix region to the intermitochondrial space. Okay, they're going to be moving towards. So intermitochondrial region, they're going to be pumped out towards there by these complexes, complex 1, 3 and 4. Okay, 1, 3, and 4 are going to be pumping the hydrogen ions because they were the 1, 2 are going to be dehydrogenazing and they were going to be producing the electrons, while the complex 2 is just they are being passed on to the coenzyme Q. So their pathway is the same, so we just remember this complex 1. While the complex 2, if the FADH2 is being oxidized, then it is being utilized. So that is not involved in the pumping of hydrogen ions. Remember this one thing: complex 1, complex 3, and the complex 4. These three are the ones that are going to be pumping the hydrogen ions from the matrix into the intermitochondrial space. Okay, once they pump the hydrogen there, because of the energy released by the moving electrons, okay, they're being pumped, and this basically there are more hydrogen ions in the intermitochondrial space, means the hydrogens in higher concentration, okay, they are in the higher concentration in the inner mitochondrial space, while they're in the lower concentration right here. So what happens is that there is this structure which is referred to as in the intermitochondrial region in the inner membrane of the mitochondria, which basically consists of an F0 particle and an F0 channel and an F1 particle. Okay, F0 channel which is going to be moving the hydrogen ions, and an F1 particle which is going to be rotating and causing the movement. Okay. So that is happening. When you discuss the structure of mitochondria, you do discuss in the intermitochondrial region is that there is a presence of some F1 particles there. Okay, that is basically the F1 particle which rotates. Okay, so what happens is that there is higher concentration of hydrogen ions here, while there is lower concentration of hydrogen ions here. So what happens is that this F1 particle, what is going to do? It is going to be through F0 channel. They're going to be moving the hydrogen ions from the intermitochondrial space into the matrix. So it is moving from higher concentration towards the lower concentration, and this is going to be utilized for the production of ATP. So ADP plus phosphate inorganic is going to be getting converted into ATP right here. So that this is going down the concentration gradient, it will be releasing energy which will be utilized by the ADP for the production of ATP. This is what chemiosmosis is. Okay, so you need to remember there are 3 steps in the respiration. We need to remember what is electron transport chain, where in the mitochondria it take place, what are the different components of the ETC, and then we discuss the different electron flow that take place in the ETC complex 1, 2, 3, 4, and the cytochrome C and the coenzyme Q. Okay, and as they're going down the concentration gradient, electrons are going down the energy level, so they're releasing energy which is being utilized for the production of ATP. And how are they being utilized? By the process of chemiosmosis, where what happens is that there is complex 1, 2, 3, 4, but in the chemiosmosis is going to be utilization of complex 1, complex 3, complex 4, because they're present throughout the lipid bilayer membrane, while the complex 2 is not present throughout the lipid bilayer membrane like complex 1, 3, and 4, and they're going to be pumping by the electron that are being there. Their energy is going to be utilized for the pumping of hydrogen ions across the inner membrane of the mitochondria towards the intermitochondrial space, and they're going to build up the hydrogen ion concentration there. Their high concentration is going to be there, lower concentration is going to be there. Through F0 channel and F1 particle, what is going to be do? F0 channel is going to cause the movement of hydrogen ions from the intermitochondrial space to inner matrix, and that is going to be utilized for the production of ATP. Okay, so here we are done with ATC. Along with that, we also discussed the chemiosmosis. Okay.